In this video, we're going to talk about cancer genes. Now, let's start by saying what they are not. Cancer genes are not extra genes that cause cancer. They are other genes in the genome that, when mutated, can lead to cancer. So, there are two classes. We have tumor suppressor genes and proto-oncogenes. And as their names suggest, tumor suppressor genes help to suppress tumors or prevent tumors, whereas proto-oncogenes serve to promote cancer. So we can think of tumor suppressor genes as the brake pedal, and we can think of proto-oncogenes as the gas pedal. And this all goes back to that cell cycle and those checkpoints. So at our two checkpoints, the tumor suppressor genes are the brake pedal. They say, all right, let's stop until we fix all of the problems with the copies of the genes. So if we made any mistakes, let's fix them. Whereas the proto-oncogenes are the ones that are trying to move past the checkpoint. So we've got the brake pedal, and then we've got the gas pedal, right? And so the tumor suppressor genes, they help control the cell cycle. And so a really important idea here is that for tumor suppressor genes, it's underexpression. Underexpression of tumor suppressor genes leads to cancer. If we don't have enough tumor suppressor gene or enough of the protein produced by a tumor suppressor gene, so essentially the tumor suppressor gene is not as active as it should be, that's when we can get cancer. Because then it's really easy for the gas pedal. If there's no brake, it's like you're going down the freeway and all of a sudden you don't have any brakes. You could go out of control pretty easily. So without the brake pedal, that protein from the tumor suppressor gene, cells can take off dividing and multiplying when you don't need or want them to. Meanwhile, with the proto-oncogenes, overexpression of these guys leads to cancer. And that makes sense as well, with our analogy to the gas pedal. If you're going down the freeway and all of a sudden you go pedal to the metal, you could pretty easily go out of control, and you're going to be going very fast. And so when the cell cycle goes out of control, that's when we get that clump of cells, that tumor, to develop. The last idea I want to touch on here has to do with recessive and dominant genes. So tumor suppressor genes are what we call recessive. And what that means is, as we talked about before, you have two copies of every gene. And with tumor suppressor genes, say you have a mutation in one of your tumor suppressor genes, and that could be bad. But since you have two copies, as long as you still have one good copy, you should be okay. So it actually takes two hits for these bad consequences to occur. Meanwhile, with proto-oncogenes, these are mutated in a dominant fashion, which that requires only one hit. And that kind of makes sense, because when you think about a proto-oncogene, even if you just mutate one copy of a gene, right? So even if just one copy is getting more gas, then we could have a big issue that could lead to cancer. Alright, so now that we understand a little bit more about cancer genes, let's see what genomics can actually do to help prevent and treat cancer. Alright, so what genomics can do. Alright, and the first thing that we're going to talk about is predictive power. So this is before anyone actually even gets cancer. The idea here is that we can look at someone's genome And based on mutations that they already have, we can predict whether they may be more susceptible to certain types of cancer. And so the one that we're going to talk about here is breast cancer. And one gene that is known to be involved with breast cancer is BRCA1. And so we're going to look at a couple different cases of BRCA1. So the normal gene, what it usually should look like when we have one hit or one mutation, and then when we have two hits. And these are all BRCA1. So they should all be the same, but they're not going to be the same. And that's because the bottom two have mutations. So maybe in normal BRCA1 we have a base pair right here. Maybe that's a C or something like that, cytosine, right? And then next we're supposed to have a guanine. And so we do, except maybe not on this strand down here. Maybe there isn't a guanine. Maybe it's one of the other base pairs, right? And so we'll continue going. And now we have another base pair. And then now we're supposed to have yet another base pair. And this time we don't. We have a problem again. Something's just not quite matching up right. So as we can see, the normal gene looks like this, and of course the gene would be much, have many more base pairs than what I've shown, but I'm just doing a hypothetical example. So maybe this is the normal sequence, and here's a sequence with one hit or one mutation, and we can see it's right here, right? Because this is where they don't match up. And then if we move to the two-hit version, right, we've got a hit there, because this two hit doesn't match up with the normal on that very last base pair, and then also on this second base pair right here. So what's the difference between the one hit and the two hit? Well, an important distinction to make is that how deleterious, or that really just means bad, I'll just put that in parentheses, how bad the mutation or mutations, because you could have multiple ones, are, depends on how many and where they are. So how many mutations you actually have and where they are. So maybe this particular hit doesn't do anything bad. BRCA is able to function normally, the protein that it produces functions normally, it's all good. But in this two-hit case, even though this mutation isn't a problem, this one right here, that's, maybe that's a really big problem, and that causes major issues, and that can lead to cancer. So that's where the mutations are, where the SNPs are, these single nucleotide polymorphisms are, that contributes to how deleterious that particular mutation is. And if you have multiple mutations, that can also be worse, because then you have a higher probability or a greater chance of getting a mutation in a bad place. And an example of this that you probably heard about in the news recently is Angelina Jolie. So Angelina Jolie, she had her, well, she didn't, I don't think she had a whole genome sequence, but she had a genetic test done where they did what's called genotyping, most likely, which we'll talk about in a later video. But essentially, they looked for a couple of particular SNPs, and based on what they found in those SNPs, it was said that she had an 87% chance or risk of developing breast cancer. And that's very, very high, 87%. So that means out of 100 people with her genetic profile, with the SNPs that Angelina Jolie had, 87 out of 100 people would develop breast cancer. Those are not good chances. So clearly it must have been a bad hit, or must have been multiple bad hits. Okay, and just to emphasize again, not all hits are the same. So for Angelina Jolie, her risk of 87% is based on the mutations in her BRCA1 gene. This is a personalized medicine analysis because someone else with the same number of mutations may not have the same risk, depending on where their mutations are. Another way that genomics can help with cancer is through what's called tumor sequencing. And tumor sequencing is just what it sounds. It's sequencing the DNA of a tumor. So on the last slide, we were talking about predicting cancer and predicting chances and risk factors. And now we're talking about treatment. So before we were saying, okay, the person doesn't have cancer, what are their chances of getting it? Now we are assuming that the patient has cancer. And now we're trying to figure out how best to treat them. So as we said, with tumor sequencing, it involves sequencing the DNA of the tumor. But before you do that, you need to sequence the person's DNA. And why is that important? Well, if you just sequence the tumor, that's great. You can figure out what the sequence is for the tumor, but you don't know what is wrong with that particular sequence. However, if you sequence normal DNA in that person, and then you compare it to the sequence of the one of the cells in the tumor, then you can say, oh, here are the mutations that are different between these two. So those mutations in the tumor are probably the ones causing the cancer. Um, Now, we'll just pull this out a little bit and really emphasize that. So once we sequence both, then we're going to compare, and the differences, any differences, should at least be looked at for possibilities for being, for causing the cancer.